Greetings to everyone. My name is Prof Teane from UNISA College of Education, the Department of Adult, Community and Continuing Education. Today's topic is on meiosis. And here is the content that we will be focusing on. We need to look into the key terms in this meiosis topic. We have a revision of mitosis from grade 10. We then tackle both first meiotic and second meiotic division. We look into the importance of this process and also how does it differ from mitosis. And finally, we look at the changes or what we call abnormal meiosis. And these are a few key terms that you need to know. Terms like cell division, which is the process by which a parent cell splits into two or more daughter cells. Chromosomes are long thread-like structures made of DNA and carry genetic information. Karyotype is the complete set of chromosomes in a cell displayed in a standard format, usually arranged in pairs and also ordered by size and shape. Humans have 46 chromosomes, which is human karyotype. A chromatid is one half of a chromosome, and chromosomes are made up of two identical chromatids. Meiosis involves the division of the nucleus and of cytoplasm. We call the division of the nucleus karyokinesis and the division of cytoplasm cytokinesis. At the end of meiosis, new cells will be formed, and we refer to such cells daughter cells. All right, let me take you back to grade 10 syllabus, where you were taught the structure of the cell, as well as another cell division, which is called mitosis. We really need this background to understand what meiosis is. From grade 10 syllabus, we were taught that there are four most important phases of division during mitosis, which are prophase, metaphase, anaphase, and telophase. Interphase, it's not a phase of division per se, but this is the first stage which informs the stages prophase, metaphase, anaphase, and telophase. During the interphase, there's no division. As already indicated, this is the stage, or this is the phase whereby the cell prepares itself for division. More food is being stored. But there's one most important thing that is happening during this phase. The DNA replicates. Remember, at the end of mitosis, there'll be two cells that are being formed. And each of these cells must have a DNA, which is identical to the mother or original DNA. Let's quickly go through these stages. During the prophase of mitosis, we have the chromatin that starts emerging as chromosomes. and some spindle fibers developing nucleus, the nuclear membrane and the nucleolus disappearing. And we have the next stage of phase called metaphase. That is when these chromosomes now arrange themselves as an equator, an imaginary line formed by the spindle fibers. In the anaphase, these chromosomes, which are made up of chromatids, the chromatids are being separated, being drawn towards the pole, and finally we have two daughter cells formed. I want us to remember this as we engage on the main topic, which is meiosis. Meiosis is a type of subdivision that occurs in sex cells or gametes. Perhaps to take you back in that slide where I talked about mitosis. One of the functions of mitosis is to bring about growth. Let me take you back in the topic of human reproduction. We all know that after the sperm fuses with an egg, a zygote is formed. And this zygote, for it to develop and end up forming the whole human being, the cells must divide. And that process of division is called mitosis. But in this case, we are talking about meiosis. It is a cell division that takes place in sex cells or gametes. When I talk about sex cells in human beings, I refer to a sperm and an ovum. When the nucleus of a sperm and that of an ovum or egg cell fuse, the process called fertilization, a zygote is formed. But bear in mind that these sperm cells and the egg cells are formed by the help of meiosis. During spermatogenesis, the formation of sperm cells, and oogenesis, the formation of egg cells, the number of chromosomes are being halved. Remember what I said? I said a human being has got 46 chromosomes. So if these chromosomes can be left the way they are, if each sperm and each egg cell has got 46 and 46, it will be 92. It will be an abnormal human being. So one important role of meiosis is to reduce the chromosome number from being diploid to haploid. And this is very essential for sexual reproduction because it allows offspring to have the correct number of chromosomes when egg and a sperm unite. All right, let's look at the diagram here. It shows a sperm and an egg. And this sperm has got 23 chromosomes, the egg 23 chromosomes to make up 46 chromosomes, which is a human karyotype. We said a karyotype is the complete set of chromosomes in a cell, including their number, size, and shape. while a karyogram is a visual representation or just a photograph or a diagram of the karyotype. I'm repeating again, human karyotype has 46 chromosomes. Some even refer to this in terms of pairs. They would say 23 pairs of chromosomes. How do we prepare karyograms? They are being prepared by taking photographs of these chromosomes and arrange them in a standardized way. All right, let's look at these karyotypes and karyogram. This is a photo that shows, let me put both of them here. If you look at the first one, the top one, it says the normal female karyotype. There are 46 chromosomes. And the below one says normal male karyotype, which is 46 chromosomes. Now the difference lies here. In the normal female, the sex chromosomes are represented by X and X. But the sex chromosomes in male are represented by X and Y. Hope you understood this. Right, let's proceed. Now we are getting to the business of the day. We're looking into the first meiotic division. Remember I said meiosis has got two phases or two stages, and each stage has got phases. The stage number 1 is called the first meiotic division, and stage number 2 is called the second meiotic division. So for now, we're looking at stage number 1, the first meiotic division. Here is the interphase. I've already indicated what is happening in the interphase. It is not a cell division as such, but this is the stage where, or a phase where a cell prepares itself to divide. There's this important thing that is happening during the interphase, which is DNA replication. So the cell is now ready to start dividing. All right. The first stage of division in meiosis is called prophase. Let's look at the diagram on your right. This is the diagram of the male cell and of an animal cell, pardon. And this is the nucleus. So during the prophase, the nuclear membrane, this dotted line, indicates the nuclear membrane that starts disappearing. It disappears. Not only the nuclear membrane, we also have a cell organelle inside here called the nucleolus. It will also disappear during prophase 1. We call it prophase 1 just to indicate it is the first meiotic division. In this phase, again, the chromatin network, which forms part of the nucleus, it now changes shape. They grow into, to the prophase. So the chromatin condense into chromosomes. So they become chromosomes. And these chromosomes are paired. It's not a single chromosome. They are paired. And we refer to them as homologous pair of chromosomes. Why this pair? It is because one chromosome is the maternal one and the other the paternal one. Each and every human being has got both maternal and paternal chromosomes. And when we talk about genetics, you will understand why. Even if you have chromosomes or genes of your father, you may not be like him, or of your mother, and you may not be like her. But you have those genes of both parents. So a pair of chromosomes emerge, and also as indicated, the nuclear membrane and the nucleus starts disappearing. The spindle fiber is being formed. There's one important thing that I would want us to remember here. If you look at this diagram here, at this point, let me first explain that each and every chromosome, this is a chromosome, is made up of two chromatids. A chromosome is made up of two chromatids. Now, when we look at this point here, there's a form of attachment, meaning chromatid from one chromosome is attached to chromatid of another chromosome. Why? To exchange genetic material. We refer to this point of attachment the chiasma, or plural chiasmata. And this process whereby chromatids from different chromosomes are attached at a certain point to exchange genetic material is called crossing over. The importance of crossing over, which is caused by an overlap which exists between two chromatids, is to exchange genetic material. So finally, at the end of this, you will see this during the metaphase. The genetic makeup of chromosome 1 and chromosome 2 is going to be different. You will see this as we proceed with other phases. That is the importance of crossing over. Right. Then we're moving to the next phase of division, which is called the metaphase. If you look at these chromosomes, a pair of chromosomes called homologous pair of chromosomes. These chromosomes are now different. They're not like the original chromosomes. Why? Because there were some changes of genes. There was crossing over where genetic material was exchanged. Now, during the metaphase, one important thing that is happening is that these chromosomes, now a homologous pair, arrange themselves at the equator of the spindle. Okay, this is what we call the spindle fiber. This is the spindle fiber. And this part of the spindle fiber is called the pole. It's formed by centriole. Here are chromosomes arranging themselves at this imaginary line at the center that we refer to as the equator. They line up at the equator as homologous pair of chromosomes, and their arrangement is totally up to chance. There's no random arrangement, there's no a specific arrangement. It is just a random arrangement of these chromosomes. And this also contributes in bringing about further variation. Okay. I'm trying to explain what I mean by random arrangement of homologous pair of chromosomes at the equator. The random arrangement of homologous chromosomes refer to how homologous chromosome pairs line up randomly at the equator of a cell during metaphase 1. So each homologous pair of chromosomes consists of one chromosome from the mother and one from the father, as already explained. So they line up at the equator in no specific order, and we refer to that as random arrangement, which leads to variation in gametes. The different combination of maternal and paternal chromosomes that result from this random alignment mean that each gamete is genetically unique. What do I mean by this? Remember, I said meiosis is used during the formation of gametes, of egg cells, of sperm cells. So those sperm cells and egg cells that are being formed are not the same, they're genetically different. And this is made possible by what we call random arrangement of homologous chromosomes. And this is one of the key reasons siblings are not genetically identical. You may wonder sometimes when you look at your siblings and you can see that you are totally different human beings. You are not identical. This is made possible by random arrangement of homologous chromosomes, which takes place during metaphase 1. Right? Another phase of division in the first meiotic division is the anaphase. We call it anaphase 1 to indicate that this happens during the first meiotic division. See what happens now. The spindle fiber will contract during the anaphase, pulling chromosomes to different ends or to opposite poles. Remember, these chromosomes were once a pair called a homologous pair of chromosomes. Now they're being separated, and this happens during anaphase. They're being pulled towards the pole. Telophase is the last phase of the first meiotic division. What happens is, remember, what happened during anaphase? Chromosomes were pulled towards opposite poles. Then the nuclear membrane is reappearing here, and we end up having also the division of cytoplasm, what we call cytokinesis. When cytoplasm divides, this cell will be divided into two. Remember, it will be two cells that are formed as an end product of the first meiotic division. Have that in mind as we proceed. And let's see whether we have mastered the content for the first meiotic division. This is the stage of preparation called interphase. The prophase, these chromosomes will be now a homologous pair of chromosomes, spindle fiber developing, nuclear membrane, neutrolas disappearing, and these homologous pair of chromosomes will be arranged at the equator during the metaphase 1, and in anaphase 1 they'll be separated. So chromosomes will be pulled towards opposite poles. And finally, we are having two daughter cells for the first meiotic division. Then we are moving into the second stage of meiosis, which we will call the second meiotic division. And we start with prophase. Why? The second meiotic division is the continuation of meiosis 1. Remember, at the end of meiosis 1 telophase, two cells were formed. Now, during prophase 2, we see the reappearance of the spindle fiber. The spindle fiber reappears, and the nuclear membrane, neutrolas, they're not here. Okay? This is the first division of second meiotic division, which is called prophase 2. The next stage, which is called metaphase 2, it is the stage whereby those chromosomes which were just lining up there in that diagram of prophase 2 are now arranging themselves at the equator, an imaginary line of the spindle, the middle line. So remember, do not be surprised when there are two cells At the end of the second meiotic division, there were two cells. So the division is continuing in this manner. We'll always show two cells. And bear in mind that what is happening in this cell will also happen in the other cell. Anaphase 2. Same thing that happened in anaphase 1, the constriction or contraction of the spindle that separates chromosomes. But this time, please note, it is not chromosomes that are being separated, it is the chromatids. Remember what I said earlier on? Each chromosome is formed by two chromatids. So during anaphase 2, the separation is of chromatids and not chromosomes. And how does these chromatids separate? Bear in mind that the chromatids, two chromatids that form a chromosome are attached at a point known as the centromere. So this part is called the centromere. So when these chromatids are being separated and pulled towards opposite pole, this happens because the centromere has divided. It is the one that breaks up and make the process of separation possible. I'm repeating, what is happening in this cell is also happening in this other cell. The telophase 2, which is the last phase of the second meiotic division, it's like telophase 1 in the sense that the division of the nucleus is completed. Now there's the division of the cytoplasm, a process called cytokinesis. So when cytokinesis happens, each cell will be divided into two. Do not forget we were working with two cells which were the end product of meiosis 1. So at the end of telophase 2, we do not have two cells but four cells because each cell divided into two. Right. Let's see whether you understood what I said about the second meiotic division. Okay, we have the prophase, which is not indicated here, but just to make you remember, in the prophase 2, the spindle is reappearing. And in metaphase, these chromosomes now, single chromosomes. It's no longer a pair, remember? They arrange themselves at the equator, and two cells are being indicated, as already indicated. These are the end product of meiosis 1. And in anaphase 2, the chromatids are being separated. This is caused by the breaking of the centromere, which attaches the two chromatids and also attach these chromatids to the spindle fiber. And finally, we are having 4 haploid cells. What is the meaning of the word haploid? Haploid means these cells have half the number of chromosomes. In terms of human being, they have only 23. Remember, the karyotype for a human being is 46. So they are having only 23 because these are sexes. One of the importance of meiosis is that this is the process that prepares the gametes or sex cells for fertilization. How are they being prepared? The number of chromosomes are being halved. So meiosis ensures that the number of chromosomes in the species stays the same over generations. And it is there to introduce genetic variation. What we have learned in grade 10 about mitosis and what we have learned today, you will see that there are some differences. For example, mitosis occurs in body cells, whereas meiosis occurs in reproductive organs. During mitosis, chromosome number remains the same, whereas in meiosis, chromosome number is halved. At the end of mitosis, there's the production of 2 cells, whereas during meiosis, 4 cells are formed. Cells that are being formed at the end of mitosis are genetically identical. They're not different. And they're also identical to the parent. But during meiosis, cells that are being formed are genetically different from each other and also different from the parent cell. There's no crossing over during mitosis, meaning the exchange of genes is not happening during mitosis, but it is happening during meiosis through the process called crossing over. Mitosis has got only 1 cell division, but we have 2 stages of division in meiosis called the first and second meiotic divisions. During mitosis, chromosomes are not paired, meaning we don't have homologous pair of chromosomes, but during meiosis, in prophase 1, chromosomes are being paired. Right. While we can see the processes, mitosis and meiosis being easy or simple, note that there are some changes that do occur during cell division. Let's talk about meiosis, and we refer to these changes as mutations. So a mutation is the changing of the structure of a gene, resulting in a variant form that may be transmitted to subsequent generations. And this is caused by the alteration of single base units in DNA, or the deletion, insertion, or arrangement of larger sections of genes or chromosomes. You will learn more about mutations while we tackle genetics. But for now, please note that changes do occur sometimes during meiosis. And for example, we have what we call non-disjunction in meiosis, non-disjunction of sex chromosomes, the complete non-disjunction, and polyploidy. Nobody controls these changes. It's just an automatic change that cannot be controlled or to be understood. So in terms of meiosis, these changes are referred to as abnormal meiosis. Where does it take place? It can happen in anaphase 1, where there may be non-disjunction of homologous chromosomes into separate chromosomes. The word non-disjunction means the failure of chromosome pairs to separate during meiosis. The same abnormal meiosis can happen during anaphase 2, where there may be non-disjunction of chromosomes into single-stranded chromosomes that are made up of chromatids. So if there's non-disjunction of chromosome pair 21 during anaphase 1 in humans, it leads to the formation of an abnormal gamete with an extra copy of chromosome 21. If it happens that such a sex cell engaged in a fusion or during fertilization, it is fused with another gamete. Because this happens most of the time in the maternal chromosomes. If such a maternal chromosome is fused, the sex cell is with such an abnormality, is fused with a sperm cell that is normal, then we have the formation of what we call 47 chromosome, and the human being that develops from such fusion is called a Down syndrome. So a Down syndrome human being or person has got chromosomes that are 47 in number instead of the normal 46. Right? Let's see what I tried to explain here, the non-disjunction. Let's say this is meiosis happening, and this is a pair of chromosomes that must separate. A mutation and error happens, they're not separated. They're not separated, and this is the end product. Another form of error, same applies here. All right, these are chromosomes, a homologous pair of chromosomes that needs to separate. The separation is okay here. But remember, during the anaphase, we expect these chromosomes to be pulled towards the pole. The centromere must break or divide, separating the chromatids. But see what happens. There's no separation here. And that is non-disjunction. It's an abnormality. It is what we call the abnormal meiosis. See what it brings now. If there's this non-disjunction, this is the same thing here. This is the homologous pair of chromosome. This is a normal separation. But if non-disjunction happens, the situation will be like this. Both of them are in one cell. There's no separation. And in meiosis too, same thing applies. Non-disjunction will happen if these two chromatids are not separated. We expect one of the chromatids to be in here, but it is not happening. In a normal human being, they are separated. And see what happens here? We also see an abnormality. These are empty and all of them are in here. So all of these are referred to as an abnormal meiosis, or errors or mutations, changes that happen. The people that we refer to as Down syndrome, having this trait or characteristic, I've already indicated that they have 47 chromosomes and not 46. And what causes Down syndrome? It is caused by chromosome 21, where non-disjunction took place, meaning chromosomes were not separated. In weak sex cells, this happens in the mother cell, in females, during the formation of an egg. So if there's one egg where the chromosomes were not separated, or chromatids were not separated, there's an error, there's an abnormality. And when this egg fuses with a sperm cell, there'll be the formation of a human being with this abnormality called Down syndrome. Let's see, how can we know that this person is a Down syndrome? If we look at his or her toes, there'll be a very wide gap between the first and two toes. Look at this finger, the little finger is incapped. The single palmar here. Their hands are short and broad. These are physical characteristics of a Down syndrome. And they normally have a small, flat nasal bridge. Their mouths are open with protruding tongue. They have a very broad neck. Their ears are very small and underdeveloped. And they are situated low on the head. Look at the skin called epicantic fold. It's different from a normal human being, the one that has got no such a disorder. Right? These abnormalities do not only happen in that chromosome 21, we also have what we call Turner syndrome and Klinefelter syndrome. These are both genetic conditions caused by abnormalities in sex chromosomes. Turner syndrome affects females. We find this disorder in females, resulting in the partial or complete absence of one X chromosomes. While Klinefelter syndrome affects males, causing them to have an extra X chromosome. Remember what I said when I was showing you the karyotype, that photograph that I displayed? I have indicated to you that the sex chromosomes for females is double X, and the sex chromosome for males is XY. So because of these sudden changes, instead of having double X, meaning two X cells that are formed where they should display the same sex cell, which is double X, a change occurs, an error. So we find only one X instead of one X in one, in another normal egg, 2X. And then same applies to Klinefelter in males. There's no separation because of these XX chromosomes which could not separate. So this abnormal male will have, instead of XY, will have 2X and Y. So an abnormal female will have only one X, and this is leads to what we call Turner syndrome. And these differences lead to distinct developmental and reproductive health issues. It's an abnormality in one way or the other. That human being is being affected. It's surprising to note that some of the abnormalities that happen during meiosis are beneficial. But that is in plants. We'll talk about it later. We have what we call polyploidy, which refers to the changes in the gene frequency and the chromosome number, altering the species at a genetic level. During polyploidy, chromosomes are different, bivalents cannot be formed properly during meiosis, so gametes cannot be produced. In animals, a polyploidy hybrid mule results when a donkey is crossed with a horse. If by any chance this horse and this donkey have the sexual interaction, they engage in copulation, and one gets pregnant, they will produce a mule. So this is a mule. It's a disorder, and then this mule that you see here is infertile, meaning these mules cannot bring or produce offspring because they are infertile. Their infertility is brought about this process of having two different animals engaging in that sexual interaction whereby an offspring is produced. So this mule will never ever have offspring because it is infertile. As already indicated, the farmers can apply polyploidy. The very same polyploidy that I said it's a mutation, it's an error. But using chemicals, they're able to make sure that they can increase production or sort of better the quality of production using chemicals to form what you call polyploidy. Polyploidy in plants leads to increased seed and fruit size. Many commercially developed crop plants are polyploid. Example, apples, strawberries, tomatoes, wheat. And they give greater yields. It is a disorder, but agriculturalists are able to make use of chemicals to cause polyploidy in plants, which give greater yields. This is what I'm talking about. This is a strawberry plant which is exposed to a chemical. They apply a chemical here, and B, due to the effect of a chemical, there's a total non-disjunction at meiosis. And then number C here, two diploid gametes fuse at fertilization, and a new plant that develops has 4 sets of chromosomes. Look how big is this strawberry. It was not produced by natural means. The farmers or agriculturalists, they applied a chemical to increase production. Thank you for your time. I hope you have gained a lot about meiosis today.